Me in welcoming Dr. Joseph Buttigieg to the podium.
Both screens? Sure. Yeah. Okay. Good morning, and thank you again to the MS Society for the invitation to come present some of my work over here. One of my students sort of brought this up when I was looking at the picture. I looked quite serious in that picture. I'm not like that anywhere close to like that. So if I scowl, it's usually because I have bad eyesight and I can't see. So if I'm looking like that at the back of the room, it's because I can't see you. Just before I get started with this, I'll just explain exactly where I come from and sort of background that way. I'm not from Saskatchewan to begin with. I'm actually from a small island nation in the middle of the Mediterranean called Malta. Moved into Toronto, the center of the universe, and was there for a while. And a position came up at the U of R to help with their anatomy and nursing program. I used to teach first-year medical students and up-year graduate students at both U of T and McMaster. So I took the opportunity to come out here to Saskatchewan. I love it out here. I got a truck. I own a farm. So, I farm and I have a truck, so I guess I'm pretty much ingrained into the Saskatchewan culture. Only problem is I'm still a Ticats fan. So I'll just make way off the table. But I digress. One of the things with MS, and we'll sort of explain about it, how we came up with this technology and sort of therapy on it, and a lot of the inspiration comes from different knowledge and different backgrounds and having this ability to integrate and to collaborate with different individuals. And that's one of the nice and attractive things about here in Saskatchewan in the first place is that ability. In Toronto, despite there being a lot of research to begin with, a lot of people are stuck in their own silos and don't really collaborate or communicate with each other. Over here in Saskatchewan, I found that to be exact opposite. So collaborating with individuals, for example, in Saskatoon, like Dr. Michael Levin, Valerie Verge, and a whole bunch of individuals. It's not just regional or anything like that. There's this open concept, and that's one of my reasons why I love it over here. Couldn't send me back to Toronto, even if you paid me. So anyways, Greek mythology and immunotherapy. I'm not sure why this is cut off over here, but I will, sorry, can that be fixed? Yeah, just sorry, 1 second here. Nope. Okay. It's okay if it's going to be too hard to fix. We just go on. Just imagine that there's words on the side. I'm sorry. I can walk through it. It's okay. We're not going to delay the other speakers here. Whoa. Okay. Not sure what's going on, but we'll go on from there. Oh, look at that. Okay, so some sort of initial thoughts on MS, and again, with MS disease, as many of you probably are aware of, it's a very complex disease. We have we try to come across and try to find some correlation and relationships with it. For example, is it a hemispheric disease? Well, we know that as you go further away from the equator, that there's a higher prevalence, a higher incidence, a higher rate of MS. In Canada, you're looking at about 300 per 100,000. So as Dr. Levin mentioned, here in Saskatchewan, especially, is one of the highest in the world. Never mind Canada to begin with. Everyone knows a couple people that have MS, and as you get closer to the equator, it's less and less prevalent. But then again, when you look at rates of MS and you compare it to like the southern equivalent of Canada, so places like Patagonia, which would be southern Argentina and Chile, or New Zealand, which would be sort of roughly equivalent in terms of latitude to Saskatchewan, it's still much less than here, right? In New Zealand, or I believe it's about 88 per 100,000 or something like that, and in Patagonia, which is southern Argentina, it is still in that ballpark, like 100,000 or something along those lines. So it's still higher than the equator, which is like 1 per 100,000, but it's still significantly less than here in Saskatchewan. So there is a trend that as you get away from the equator that there's a higher incidence, a higher rate, but it's not really directly tied into where you are on the world. Now, a lot of my research deals with spinal cord injury, stem cell therapy, and that type of thing, but MS and spinal cord injury actually have quite a bit of similarities between the two of them. One of them being that in spinal cord injury, it's not so much the destruction of the neurons, but it's the destruction of the myelin. So when you destroy the myelin sheath, then you end up with an ability to communicate between the top and bottom parts of the spinal cord, and you end up with paralysis. So there's a lot of similarity when it comes to disease progression and disease pathology, if you will, between those two. Now, what are the theories that cause all of that in terms of MS? And the question is, it's a little complicated. But what I'll talk about is the immune system. So how does the immune system act to protect us, to protect us from pathogens? How does that work normally? And when we think of MS, we think of it as a neurological disease, but in reality, it's a blood disease when you think of it. It's the immune system doing something, and it's attacking the nervous system. Again, I apologize for this. It wasn't like that. So I'll just walk you through this over here. So the way that your body protects yourself from like viruses and bacteria, like it has no way of predicting what you're going to be exposed to. It just makes an assumption. And so what happens is your body creates a bunch of cells that randomly recognize different shapes. So recognize, for example, a triangle versus a square versus a rectangle, and just recognizes different shapes. And that's how your body generates these immune system cells. But what happens if a square-like protein is similar to yourself? Well, you don't want that to happen, and the body has an inbuilt system to prevent that from happening, and it's called a selection pathway. So what happens? Let's see. So what happens typically over here, if you have your immune cells, you have a whole bunch of generated immune cells that are being created. What happens is the body has to select out the bad autoimmune cells. So these immune cells, we're just going to sort of pour on both sides here. So these bad autoimmune cells are the cells that would recognize your own body. And you have an organ in the body called the thymus gland, and it's located just above your heart. And what happens typically is those cells are challenged. What they're told is, okay, do you recognize the body? And if they do recognize the body, what happens is if they recognize the thymus, then they undergo cell death. So you eliminate those cells. Okay, it prevents autoimmunity from occurring. And then the ones that do not recognize the thymus gland, they go into circulation, and they just wait out in your body, waiting to see if a pathogen that matches that shape specifically comes into your body and then it attacks it. So how does the body recognize what is bad? Now, over here is just a schematic diagram of a bacterial cell. You'll have antibodies that recognize what we call an epitope on this protein. So you'll have a protein, what we call an antigen, and on that antigen we'll have these little epitopes. So these little areas that your immune system would typically attack or recognize as being foreign. So you can have multiple cells attacking this particular cell or this particular protein and then destroying it. Now, what happens usually is the way that it's initiated is two pathways. One is phagocytosis. What happens over here is an immune cell sees something that's foreign and then eats it up and then becomes a presenting cell saying, hey, look what I found over here. And it's advertising to the immune cells that I found something strange. What is this doing? So what would typically happen is you have a phagocyte, so it's macrophage or something along those lines, and it engulfs a bacterium or it engulfs a virus. And it says, hey, look, I found something, and presents it on its surface. Now you have a special type of T cell, and that's the cell that says, okay, is this foreign or not? And what it'll do is come and it'll bind with that, And once it binds with it, it'll determine, yes, is this a bad cell, or are you attacking something that's part of the body? Now, if it's, yes, it's a bad cell, or it's a bad virus, or it's a bad something or other, what it does is makes copies of itself, and then those drones, those clones, if you will go out into the rest of the body and find those viruses and destroy them. Now, what typically happens, what normally happens is if that protein that the cell picks up is part of the body, it's part of yourself, and this T cell finds that, what it says is, okay, no, bad macrophage, don't eat that, that's bad, and then tells that macrophage or that cell to die. So it prevents that autoimmunity response from occurring. So the linchpin, the key to your immune system are these T cells. They make the determination whether to attack something or to not attack something. And the reason why they're that linchpin is that they've been tested by the body when they were created to make sure that it doesn't attack the body. So you'll have your antigen-presenting cells, your T cell becomes activated, and then it goes on and attack. Once it's been saying, okay, yeah, this is something foreign, goes on and tells other cells to, form an immune response and then fight off that viral infection. Now, if you mutate or affect those T cells, then you change the ability for the immune system to do its thing. So a good example of that would be HIV. I think everybody's heard of HIV, right? It causes AIDS. What HIV does is a virus that attacks specifically these T cells. If I take out these T cells, well, I have all these other cells that can form an immune response, but because these T cells don't turn on these other cells, then you don't have an immune response, and that's what AIDS is. Now, when you look at MS, what happens, and again, we're not quite sure why, is these T cells that are here are the ones that say, hey, yeah, no, myelin's bad, go ahead, attack it. And you end up with MS as a result of that. So that's the basis behind the immune cell. Now, these T cells, in turn, tell other cells, such as B cells, to produce antibodies, and that's where these antibodies come along and start attacking the myelin sheath. So you'll have these T cells that come along. They, for whatever reason, recognize myelin as being foreign and give permission to these other cells, such as B cells and such, to start producing an immune response specifically against against the myelin sheath. So why does this happen? Why do they, if they're being selected for, if they're being tested by the thymus gland, why are they attacking all of a sudden the myelin? Why does this happen? Well, normally it's recognizing foreign objects as being foreign, but there's some hypotheses that, a previous infection, so you'll have a virus or a bacteria that has a shape that's not quite like myelin, but close enough. And what happens is you'll have a T cell that recognizes that virus or that myelin, or that, protein, and then because it's so close to similarity with myelin, then it starts to attack the myelin. So that's hypothesis as being one. It could be genetic as another option. So some people or some groups of populations have a higher risk, a higher incidence of developing these T cells that recognize myelin for some reason. Again, we're not quite sure how or why that occurs. And what does it have to do with Saskatchewan specifically? Again, the jury's still out. There's a lot of research that's being conducted on this, trying to figure out what the cause is for that MS, or what's causing these T cells to start recognizing the body as being foreign. So I'm not going to belabor the point on this one, but we have the cell, we have neurons. What we have over here is the cell body. You have your axon, which communicates to another cell and forms these neural connections. So this is what forms your thoughts. This is what forms your memories. This is what forms your reactions to environmental stimuli. Okay? And so what happens typically is you have your cell body, the neuron forms an action potential. It sends an electrical pulse down this axon, and that electrical pulse tells this other neuron to do something, and so on and so forth until you get a response, right? And that's where your memories come from, and these neural connections. When we think of the brain is a very complex structure. We think of thoughts as being simple, and we do it all the time. But when we think of it, right, the brain has something like 100 billion neurons. So these 100 billion neurons are communicating with each other all the time, and they're doing this communication. Now, what happens in MS, unfortunately, is that you start to destroy this myelinated axons. Now, the context of what these myelinated axons would be sort of like a rubber coating around an electrical wire, right? You're preventing that electrical wire from shorting things out and that type of thing. So that's what that myelin sheath is there for. Now, because you're forming these immune cells that are destroying that, once you start to destroy that protective myelin sheath, then not only is the communication from this cell body going down to the next cell impaired, but on top of that, you're causing adjacent neurons. So if there's another neuron out here somewhere that's beside that, you can short out those two neurons. Remember, you have 100 billion. You have to think of that number. It's a big number stuck in this small object sitting on top of your neck. And what having these neurons close to each other causes them to become damaged, as Dr. Levin mentioned in a previous talk over here. And then eventually what they do is they start to die. Now, one of the classical sort of phenotypes, if you want to think of it of MS, I think I was just mentioned in the previous talk, is that there's this cycling between active and remission. Okay, and there's different phases. Like you have, for example, the benign phase where you have some active impairment and then it regenerates and then just keeps cycling back and forth. Now, why there's this sort of differences between these different phases or different types of MS, and again, we're not quite sure about that. But the neat thing about it, for many of these, is that you'll have an active phase and then you'll have a phase of remission. What causes that remission, we're not quite sure. But what happens during that remission phase is the immune system is shut down. So those autoimmune cells, for whatever reason, are attenuated. They're stopped from doing what they're supposed to be doing. If you do that, then it gives the body now the ability to repair that damage. And that's why you have this return to normal. Everyone here has stem cells. Your stem cells are able to repair your body, regardless of your age. Okay? What happens, though, with MS is that your body has, steady state, you have disease. Those T cells now destroy that myelin sheath. Then for some reason, it gets shut down. And then your stem cells come into play and repair that damage. And then I return again, and then go back, and so on and so forth. The hypothesis that our lab's sort of testing with this is like with the progressive stages over here is that you have a reduced stem cell pool. And as your stem cell pool sort of disintegrates or diminishes, reduces, then you don't return to baseline anymore and you end up with progressive deterioration of disease. So in terms of current therapies, and there's not really many that are out there, and this is what it leads to, what we developed in our lab. We either suppress the whole immune system. Well, you suppress the whole immune system, that's not ideal, right? You end up with basically AIDS, if you want to think of it that way. You're sensitive to everything in the environment. Filtering the blood, trying to remove these T cells, you're not going to be guaranteed that you're going to remove all of those bad T cells. There's the nuclear option, which I call, which basically you nuke the whole immune system, you start from scratch again. Again, and not the most ideal is when it comes down to. So the one thing about a lot of these treatment options is that they don't really treat the root cause. The root cause of this is those T cells. How can you eliminate specifically just those T cells? Those are what's causing the disease at the end of the day. So our idea is to give exactly what the immune system wants. It wants myelin, or it wants some particular protein, give it myelin. Sort of the idea behind the Trojan horse approach. For those of you that are not into like Greek mythology, I love ancient history, by the way. One of the things happened, or has everyone heard of the Trojan horse? I'm assuming to a certain extent. Sort of the synopsis of the story is the Athenians were attacking the Trojans. They couldn't break through the walls, so they came up with a neat way of attacking them. They made this decoy. The Trojans loved horses, so they made a big horse, and then you hid a bunch of soldiers in it. Well, the Athenians took off, hid behind a couple of hills, and the Trojans woke up the next day and look, big horse, this is what we want. Drag it into the city. Well, what was hiding inside the horse? Soldiers. So when they were partying at nighttime, soldiers came out and destroyed the city and burnt it down. The idea is now to do the same thing with these immune cells. So how can we force these immune cells now to actually take up this protein and then commit suicide is basically what we want them to do. So key to this sort of protein complex, this drug that we developed, is something called an internalization sequence. It comes straight from nature. I think everybody has seen this at some point. If you see a tree, if you see a tree, it'll have this big ball around it. Or if you have roses, and sometimes your rose petals will curl up and it'll have like this red, we call it gall on it. What happens over there is you have bacteria, and they put in a protein into those cells, and it changes the plant, the leaves, to curl up around that protein and protects it from the environment. So the protein manages a way to sort of trick the cells into doing something it wants. Now, we do this in our stem cell therapy, at our stem cell trials, where we cause stem cells to pick up a particular protein and then change into something. So I want to take a stem cell, turn it into a neuron, or turn it into an oligodendrocyte. We use this particular internalization sequence to do that. Now, sort of a thought that happened, again, like as I mentioned at the beginning, I could farm. So I'm seeing this crown gall. I was like, I wonder if we can use this for MS. And the idea basically is to tie it in with a whole bunch of other proteins here. So we give the immune system exactly what it's looking for. We give it myelin. It's looking for myelin. We also give it that internalization sequence so that anything that binds to that myelin picks it up. Once it picks it up, there's a little bit of a death cue attached to that, causes that cell to commit suicide. So by that way, we're targeting specifically the cells that are looking for myelin. I'm not attacking any other cells. I'm not attacking the cells that are protecting you from the influenza virus. I'm not attacking the cells that are protecting you from, bacterial infection or something like that. We're not taking those cells out of commission. What we're doing is a very targeted strike, and we're taking out specifically the cells that are causing the disease. So to give it an idea, using Pacman, so yes, I'm an 80s kid, so. What happens over here is you have macrophage, or you have a B cell or a T cell that's specifically looking for myelin. And what happens is, it's eating that myelin, it's doing that autoimmune response, and you end up with MS as a result of that. Now, if I take this complex, well, it looks like myelin. Is it looking for myelin? The answer is yes. And so, eats it, and it destroys that cell specifically. It doesn't take out any of the other cells that are involved with this. So it's game over for that mutant immune cell. The nice thing about this is it doesn't work specifically for MS. It could theoretically work for a whole bunch of other autoimmune diseases. So things like myasthenia gravis, Lambert-Eaton syndrome, Goodpasture syndrome, there's diabetes 1, or type 1 diabetes. It'll attack any of those as long as we give it that special construct to tell it specifically what to attack. So, sorry, I was hoping that this would have worked out nicer with the sizes. But anyways, with funding from SCHRF and the collaboration with Dr. Levin's group, what we did was develop a mouse model of MS. It's called experimental autoimmuneencephalopathy. Can you see that like really fast 3 times? EAE is the best way to say it. And what you can do is you can simplify the disease. And again, with MS, it's a fairly complex disease. There's many antigens and epitopes involved with it. You simplify it to just one epitope, one antigen, and see if it actually does anything. And so what happens over here is we can give these mice basically MS or MS-like symptoms, and then test our compound to see whether we actually neutralize those autoimmune cells that have been generated inside that mouse. I'm happy that this one worked because this is where our data comes into play. Now, at the University of Regina, we have the ability to count cells, to actually see what types of T cells, these B cells, the defective ones at least, are present in that mouse. And so what we're looking at, and bear with me here, so on the y-axis, we're looking at basically numbers of cells. On the x-axis over here, we have our different treatment groups. So it's shown on both sides. So we have our y-axis and our x-axis showing the different treatment groups. And we're counting how many cells there are. Now, when we have our no treatment control, okay, so we gave them EAE, they haven't been treated with anything. And you can see that their immune cells, their T cells and their B cells are quite elevated compared to your healthy control. Okay, so these are animals that are perfectly normal, no problem. What happens though when we treat these cells, or these animals now, is we have a decrease in the number of T cells compared to your EAE treatment. So you take a look at this animal that has MS, for lack of better words, and you look at our treatment groups, our 2 treatment groups knocks that down significantly, our T cells. We're specifically knocking out these T cells that are causing the problem. We take a look at our B cells, and it's the same thing. Compare that against your test group, and what we do, well, we knock that down significantly. So what ends up happening now is what we're seeing is that we're targeting these immune cells, and we're specifically removing them that are causing the problem. We're not wiping out the immune system because it's not down at 0, but it's bringing it down significantly. So, well, that's fine and dandy. We looked at how many cells that are in there. What's happening to the whole animal? Well, you have the ability with the EAE model to actually score these animals and see exactly where they come on like open field, so how they walk. And so you can see what ends up happening with progressive severity of MS in these mice is that their tail will start to go limp. And then after that, it'll work to their hind legs, and the hind legs will start to go limp. And then after that, it progresses further and further to the front of the animal, and then eventually front limbs will become limp. And then what will typically happen is the animal will sort of fluctuate, exactly like the relapse-remitting MS type, and they'll fluctuate between active phases and reduced phases, or remission phases of MS. And what we find over here, and this is just the last one that we did this past month or so. And what we're looking at, again, is the different treatment groups. And what we have to take a look at is what's happening over here at day 23. So day 0, the animals are perfectly fine. There's nothing wrong. And then roughly at about day 12-ish, then they start to get progressively worse. At the peak of where they are the worst, so to speak. So at day 15-ish, day 14, we give them their treatment. And as soon as you give them that treatment, it starts to reduce the severity of MS. And what happens, which is kind of neat, is that it brings it down almost to baseline. So they are paralyzed at first, and instead of cycling like a regular mouse would, so sort of this green line over here would just keep cycling back and forth between active and remissive phases, we actually put them in permanent remission. Okay, now to hopefully, if this video works. Okay. I am not sure. Nope, okay. Can you click on that, or? I'm not sure how to do that here. There we go. Okay, so what we have over here is a mouse that has EAE. This is Anastasia that's doing this experiment. Now, what happens with this mouse, you can see its back legs and its tail are dragging around. Okay, so this is just prior to us giving it any treatment or anything like that. So it's not doing that great, unfortunately. Now, at this stage, we take this mouse and then we inject it with our trimeric protein. Sorry, if you can click on that. And this is about 8-ish days or so afterwards. So the mouse is walking around. And from what we can tell, at least, it puts it in sort of permanent remission. So the animal is able to walk around. There's no sort of return to MS or MS-like symptoms. Now, that being said, we haven't gone beyond about 14, 15 days post-treatment to see, because we're just seeing, okay, does it work in the first place? No sense doing a 3-4 month trial if it doesn't work in the first place. So we tried this already now about 3 times, and we're seeing the same thing over and over and over again. It's very significant is what it comes down to. So the neat thing about it is that, again, sort of recap, is that we're specifically targeting these immune cells that are causing the issue, right? If you can target that and remove the problem cells in the first place, it gives the body at least the ability to repair that tissue if there are stem cells present. And then if not, if no stem cells are present, that's sort of the other area of my research, is to inject stem cells and then help the body then repair itself. Because it doesn't make sense in the first place, right, for me to inject a stem cell into a person that has these T cells. Those T cells are still going to destroy what I just put in there. So if you can remove those problematic immune cells, then, well, you can start doing stem cell therapy if that's required. My hypothesis is if you're early enough stages of MS, you wouldn't need that to begin with. Now, mind you, that mouse isn't 100%. The tail is still limp, but at least there's a better quality of life, if you want to think of it that way. There's an improvement. It's actually mobile and such. So we think it's fairly promising. So future directions. Well, currently we're moving to animal systems, to test the efficiency, the toxicity, and the data, as you can see, is quite promising. We want to do a dose response curve. I guess I can go back a couple slides and then show that, but there seems to be a weird effect that's happening between a 100 nanogram and a 200 nanogram dose. We're not quite sure what's going on over there. So you have to sort of tease out a couple things is what it comes down to it. The other thing about our study and our study design is that we simplified MS. We simplified it to its basic elements to see, just a proof of concept. Now, the next idea is to make it more complex, to see if we can add in multiple epitopes, multiple areas to recognize and see if it works in that. So that's basically what it comes down to. Now, I'm going to sort of do a little bit of a sales pitch over here and sort of an advertisement for a couple things. One thing that I truly believe in, being a researcher and being a relatively young researcher at that, is sort of flipping the lab and doing things differently. A lot of it, what it works out to when it comes to like research, is you have, an egghead like myself develop a drug and then send it out, and then that's it. And then maybe it works or it doesn't. Sort of distance from the community in general. What I'm hoping to do is start recruiting patients to actually be embedded and actually help with the research itself. You don't need to have a science background or anything like that. But it would help to give a perspective, at least to myself and to my research group, saying, okay, these are the things that as an MS patient we'd be looking for in a drug or in a treatment. Helping me with, ethics and recruitment and that type of stuff. So, in having the patient not as sort of a knowledge user that's, out there somewhere, actually have the knowledge user actually as part of my research team. So you're actually working with and alongside with us, and you can actually see where research funds goes into. You can see where research is going and heading for that matter. Then another thing, and I was sort of thinking about this, that people were asking me, well, when are you going to start clinical trials? Well, the ubiquitous answer by every researcher is, 7 to 10 years. This is what they all say. And it's horrible, and it irritates me, because you don't know as a researcher. You can take a drug and you can turn it into, effective treatment in a couple years, or it can be 2 decades or more. And that's not exactly viable in what way I want to do it. But the neat thing about clinical trials, and you're going to hear about this probably a little later from our keynote speaker today, you typically have to have a lot of animal work already done and that type of thing. Well, what did I say about MS? MS isn't a neurological disease, it's a blood disease. So what I want to do in the relatively near future, we're talking about within the next year, is actually take blood samples from MS patients and then do this exact same experiment, but on blood, just do it in the lab. We can count the cells. We don't have to actually test it on the person. What we're curious about is, can we eliminate those problematic cells? Well, we can do that in a petri dish in my lab. That's not a problem. And blood samples, well, everyone's donated blood at some point, and I'm not looking for much. It's 25 mils. It's basically a couple tablespoons. I should have done this around Halloween. I'm looking for your blood, please. No, but what we're hoping is that in about a year's time or less, is that we can start recruiting patients and actually doing these experiments specific on patients' blood. Now, when we go to Canada Health and say, hey, we want to try a clinical trial, they want to see everything. We say, oh, yes, but we already tried it on humans. Not injecting it into humans, but we tried it on humans nonetheless. So that's sort of one of the future directions that we want to do. So can we sort of skip some of these steps in drug development and drug trials and actually implement it and go from there? So relatively near future, we'll be asking patients as well for blood samples to test on, right? So 2 things that sort of summarize what we're looking for. Patients to be incorporated with our research program, and then also for patient blood samples who can actually do these sort of more advanced complex tests on humans for that matter. So the neat thing is sort of the tie-up for all of us is that this treatment isn't just for MS. Now, we focus on MS because I have a very sort of close personal connection to that. I have family members that have MS. I have friends that have that as well too. So it's a bit of a close personal tie-in with regards to that. But I also have friends and family members, for that matter, that have also other autoimmune diseases. Things like type 1 diabetes, Crohn's, celiac disease, myasthenia gravis, Goodpasture syndrome. Like, there's all these autoimmune diseases. This theoretically could work for any of them. But right now, we're just focusing on the MS component and seeing that goes from there. So, without further ado, sorry about the technical complications here. I do apologize for that. I'm going to blame it on the weather outside. But I'd look forward to any questions that you may have. Yes, sir.
What's the difference between one of your plaques and a Lewy body?
So what is the difference between?
One of your plaques and a Lewy body.
Well, when I'm not looking so much for plaques and Lewy bodies in this context over here. I'm not so much interested in what's happening to damage-wise to the nervous system. I'm looking at elimination of what's happening with the immune system. Now, the effect on the nervous system, so plaque development and that type of thing, I haven't looked at that, so I wouldn't know.
Okay, if you would raise your hand if you have a question. There's 2 of us with microphones. We'll try to get around to as many as possible in the time that we have. And you can see the other one over there? Okay, so we're going to start right here. And if you can't hear the question, just ask us to repeat it.
Okay.
Hello, my name is Russell, and I am curious if you've done any research with, say, mice where this has been allowed to progress a while, and then do they have any recovery after that?
So you're talking about more so like with like a chronic phase and such, right? Or more advanced phases of MS? And the answer is we haven't done that. We haven't progressed that far. What we've been focusing on is just the acute phase. The assumption, though, just based off of our model and disease progression, is that it should work on the chronic phase. Because again, whether it's chronic or acute, what you have to do is prevent those immune cells from doing what they're doing, and it's not what you want, right? And so the idea is, the thought is, or the hypothesis is that if you eliminate that problem cells, that it should have the same effect. Now, in chronic phase, the question is, do you have the stem cells to repair? We might be able to stabilize you, might not return you back to normal, but at least you're not going to get worse. And then this is where the stem cell therapy comes into play, where you can do an injection of stem cells now at this point, and then bring you back down to where you were. So, yeah.
Okay, we have one over here.
Hi, my name is Triss. I have a question. I see that everything is kind of focused within our human body, and that's where you're looking for the majority of the answers, and I can see that. But I actually think there might be more to it than that, because I'm wondering, when Dr. Levin spoke about the hemisphere and stuff, I think geographically it really showed something when he put the actual thing up showing all the red dots and the location of those dots. And that really makes me wonder, well, why is it so high in Saskatchewan? And, is it maybe the answer isn't within us, perhaps it's out there. And maybe that's where we actually might find more information that can help us.
Oh, I'm not disputing that. It's probably something from the environment. What you're talking about, okay, so what I'm talking about is one step separated from the cause of MS, right? You have these T cells, they're the ones that are causing MS. They're the ones that cause the problem. What you're asking is what causes those T cells to do that? And the answer is we don't know. Now, not to say that I'm not going to say that it's irrelevant or a moot point, but as long as you get rid of the problem cells, what causes that? It's more so of a question and exercise of scientific inquiry. I'm definitely interested in that. Don't get me wrong, but I'm also interested in, okay, if I get rid of that problem cell, well, then I can go downstream from there. So I'm not disputing what you're saying, that there's something, it's more complex. There's an environmental. There's probably something to do with the sun. We think-ish, maybe genetics and like background ethnicity, probably. Like in my home country, MS is unheard of, right? There's like one, but mind you, there's like only like 400,000 of us. So, but there's like 1 per 100,000, so like 4 on the island that probably have MS, right? Like it's very rare. Is it something to do with Saskatchewan? Possibly, but then why not Manitoba or Alberta, right? Maybe it's a Rider Nation thing. Go Ticats.
I want to clarify a few things. So a Lewy body is different than a plaque. So a Lewy body is a neuron in someone who has a disease that looks like Parkinson's disease. And in that neuron are protein aggregates, large proteins that get stuck, they can't dissolve. That's a Lewy body. It's not surrounded by a lot of inflammation, maybe some macrophages. It's different than the MS plaque, okay, which again, there is neurodegeneration there, but the plaque is filled with B cells, T cells, macrophages that attack. Now, where might they come together? When a nerve cell dies, slowly over time, they might both involve stress granule formation. So they're associated with different diseases, Lewy bodies and the MS plaque. They look different under the microscope. We think they form in very different ways, but they may end up killing the nerve cell in the same way. So they are different that way. I wanted to clarify that. Environmental factors are complex. Identical twins. So identical twins have a 30% chance of being concordant. That means if twin number 1 has MS, there's only a 30% chance that twin number 2 is going to have MS. To me, that says there's a 70% reason, that's not human, that there's something in the environment that's triggering this. And certainly, part of our research here is to figure out why this is happening in Saskatchewan. And stay tuned, because we'll be looking at those. Yeah.
Another question?
Is okay? Can I go? So I wanted to check a question you had about like the Trojan horse system that you're using. And
Sorry, I couldn't see who was. Yeah, sorry.
Sorry.
I'm looking around. Sorry, I apologize.
And the myelin sheath, so that you're inserting myelin into the Trojan horse for the T cell to eat. What is your source of the myelin that you're using?
Well, I'm using sort of simplified terminology for it. It's called MOG, so myelin oligodendroglial protein. We'll just say myelin. And the source of it is from other mice. So it's a MOG peptide. It's similar to myelin. Like I said, we sort of simplified this immensely from the complex antigen that's there. So it's myelin from another mouse. We use that myelin to induce the MS response in that particular mouse. And then we use that myelin now attached to our peptide to see if we can eliminate those cells that we induced in the first place. So does that answer your question? Well, you can use human myelin, but the effect would be, it should theoretically be the same, because in terms of evolutionary conservation between human myelin and mouse myelin, there's not that much difference. So in the context of doing our therapy on, for example, human blood, in that case, we have access to human myelin, and we would do that.
Are the T cells specifically targeted?
Sorry.
Which of the T cells are specifically targeted, because there are many of them, as you know?
Well, definitely. So we're not so much just targeting the T cells, but it's also potentially targeting B cells, as you saw from our
Or the
figure.
B cells, yeah.
So yeah, because with both those cell types, what they're recognizing, and again, for lack of better words, make it simple, is they're recognizing a particular shape that's unique to myelin, or in this case, myelin oligodendroglial protein.
So the T cells are from the thymus, and if you eliminate the thymus by surgery, it helps out in myasthenia gravis. Has there been any work done in that regard?
For MS,
Yeah.
typically no, and unless Michael Levin
Theoretically,
thinks,
it would work.
well, sort of go ahead.
So the question is a thymus, which is the immune organ right under our chest, filled with mostly T cells and some B cells. If you remove the thymus from myasthenia gravis, which is a muscle disease, patients tend to get better. Has it ever worked for MS? I don't know if it's ever been used for MS, but part of the issue, the difference between myasthenia and MS, Walter, as you know, the immune response gets stuck in the brain, right? And it's hard to get those cells out of the brain. In myasthenia, the blood and the immune cells are in the periphery, and they're in the blood system in the muscle, so it's probably a little bit different. But I don't know whether in people they've ever taken the thymus out. Actually, so Walter's a researcher who's been here for decades. So I'm going to ask you. You might know more than me. Has anyone ever tried to take the thymus out? Dr. Hader, everybody. Yeah. They never did it.
The one thing about the thymus gland, and again, not to go into the anatomy and physiology, as one ages, so it starts to atrophy and it starts to disintegrate. And so, again, when you think about risk to influenza and other immune diseases, right? What are the greatest sort of risk factors? Age, under the age of 2 or over the age of 60, right? And so we're looking at those two, and the reason why is as you get older, it doesn't work as effectively. So if you removed it in an older individual in the first place, I would hypothesize, I would guess that it probably wouldn't have an effect.
Hi, my name is Danny. I'm wondering, is it an injection under the skin or intramuscular?
It's a subcutaneous injection, so it's just underneath the skin.
And would you do it, like, what are you looking for? Is this something you want to do? One, like you don't know any time limit yet on the injections? If you're,
Well,
like, what do you
so the
do on
initial idea
mice?
was to do it IV, okay? But what we selected for was to do it subcutaneous. Well, what ends up happening is it goes into, there's a fluid that surrounds all your cells called interstitial fluid, and then that goes to your lymph nodes. And this is typically where you're going to have your immune cells to begin with. So if we do it IM or subcutaneous, so IM is, intramuscular, or subcutaneous, then we're introducing it to the lymphatic system and then attacking the sort of T and B cells at kind of their source type of thing. Now, we also had another toxin called pertussis toxin, sort of permeabilizes the blood-brain barrier and allows it to easily transit from your blood and circulatory system into the brain and hopefully eliminate those cells from the central nervous system. So it's not so much, yeah, if that answers your question, sorry. Okay. Sorry, yeah.
I've been doing a bunch of stuff with animals and humans most of my life. The guys at Max Planck figured out that if you affect the zygote up to the foreblast stage, it'll skip generations. It's kind of like the morphic resonance effect. If you hurt a rat in World War II and you start the same experiments in Australia, 20 years later, they'll automatically pick up that 100 monkey syndrome. So it may be the point of action that's very hard to detect, which is why the common point in the process is when it occurs. And then down the road, in regards to the plaques, there's in chronic wasting disease something called a prion, which is a very difficult thing that occurs. And what happens with the prion is that the prion lives beyond the life of the host and then is totally infectious again. So there's a cycle in there that's very hard to control, which is both micro and macro. And that may explain some of your difficulties in pinpointing what you're doing.
When you're looking at, but a prion disease, again, is different when you're talking about an infectious agent, and yeah, it's most definitely an infectious agent. But a prion disease, again, to think of it and to sort of explain to the rest of the public over here, a prion disease, you have a protein that's mutated, in a sense, and it causes other proteins that are similar to it to become mutated. So that would be for things like, Creutzfeldt-Jakob's disease or, chronic wasting disease, as you mentioned, right? So Creutzfeldt-Jakob's would be a human equivalent of that as opposed to chronic wasting, which is you find in deer. Now, is there a transspecific evidence for that? And the answer is probably not. The only one that would be remotely close to that would be Alzheimer's, and it has nothing to do with MS. It's a completely different disease. Well, but that's not really, in this context over here, I'm just focusing on MS. So that, for dogs in South Africa, yeah, that one I'm not 100% certain of, so I haven't heard of that study. Okay. Sorry. Yeah, no worries. Yeah, I haven't heard of that study specifically. Sorry. All right, thank you.
Thank you so much, Dr. Buttigieg. And are you here for a little bit today? You're here all day. Excellent. So, and I do want to thank many of our presenters have availed themselves for the day. So if you're on a break and you have an opportunity to chat with them, please feel free to do so. So thank you.